Transcription translation is today's lesson. I put like an overview picture, so just to show what's going on. Here's the DNA in the nucleus. So we're in a eukaryote. You can see the nuclear pores. And we're just going to look at, put a little asterisk here. So we're going to say what happens in this part of the nucleus. This is where transcription occurs. And then over here is translation. I made a little diagram just to kind of start us off. So let's get into it. I always, again, they're unlikely to ask about both transcription and translation in the same question. So it's important that you know which is which. And I literally think that C comes before L in the alphabet. Well, I know it does, but I know transcription comes first, so it happens in the nucleus first. So I made a little diagram here. Let's just label this. So here's some DNA. It's already, I'm already on one strand. This is the antisense strand. So yeah, it's already been unwound. I mean, it doesn't unwind to this extent. This red blob, which is the enzyme RNA polymerase, has a helicase portion to it. So as it moves along this antisense strand, it kind of breaks the hydrogen bonds between the two strands to open up a little segment of it so that it can expose the bases. And it will spontaneously kind of like, as RNA polymerase moves along, so it's moving like that way, it will spontaneously, the DNA will reform behind it. But I'm not showing that just for speed. So RNA polymerase moves along the antisense strand of DNA at the site of a gene. And these T's here aren't T's. In fact, they definitely are not T's because there is no T in them. These are RNA nucleotides, not bases. They are nucleotides, RNA nucleotides, and they can have adenine as their base, they can have guanine as their base, they can have cytosine as their base, and they can have uracil as their base. They never have thymine as their base. So this little T shape is a bit of sugar phosphate. It's the ribose and phosphate ion, and then the bases are sticking off. And on our DNA strand, we're going to get exposed bases. So there's going to be a T here, an A C, a G, a C, a C, a C, etc. So when I write about this, I've already got 2 marks saying RNA polymerase, tick, moves along the antisense strand. The key thing is that when these new bases come in, they do so, they bind to the DNA strand spontaneously. It doesn't require the enzyme. Yeah. So I would talk about, that this complementary base pairing, comp base pairing takes place. Yeah, between exposed bases of DNA and the bases of the RNA nucleotides. Yeah, and hydrogen bonds form. Names of bonds get marks. Name your bonds. Okay, and then I would give an example. I would say, and I will point out the uracil problem. And don't say A's and T's and things. Say that, a nucleotide with uracil as its base binds to expose adenines. Again, they are named in the syllabus. You have to name them, and knowing that there's no thymine is often a marking point. I'm just going to come over here slightly just for a bit of space so I don't cramp below. And then I can name the name of the bond that RNA polymerase makes. I can say that the RNA polymerase, again I'm doing it shorthand, forms phosphodester bonds. To form, well, I'm in a eukaryote, so we call it pre-mRNA. Okay, so that is transcription. RNA polymerase binds to the antisense strand of DNA at a gene. 3 nucleotides drift in. and they do complementary base pairing, forming hydrogen bonds, where there's an exposed adenine, a RNA nucleotide with uracil as its base will bind, and then RNA polymerase comes in like an iron and forms that sugar phosphate backbone bond, but it's called a phosphodester bond, and we're forming pre-mRNA. So that is transcription. Again, we do a lot more of it about it and how this is controlled in topic 8. So that's the asterisk done. Then something happens here. This arrow here, this is where splicing occurs. Again, it's not part of transcription or translation. It's the bridge between the two. And this is where mRNA, mRNA is formed from pre-mRNA. Yeah, by removing introns or exons are joined. So yeah, splicing occurs. I don't need to know the detail. So this molecule here is mRNA. Yeah, it's now been spliced and it's going to diffuse out of a nuclear pore into the cytoplasm. So here's our cytoplasm. That's our nuclear pore. So the type of things I'd write about in an essay, all this stuff about nuclear pore and things, because it's a valid bit of detail. So then we get to this. So this is our mRNA. It's just stretched out now. This is a ribosome. And off the bottom of the ribosome, I'm just going to change colour to blue. This is a polypeptide. So you can see that the ribosomes, they move along the mRNA, and the polypeptide is getting longer. So it's short here, it's longer here, it's longer here. So I now know the direction of movement, that my ribosomes are moving that way. They're moving to the left in this diagram. Okay, so we're going to get to that in a minute. So we've understood this. Then here, again, it's completely out of scale. It is a diagram. It's trying to show you that there's a cycle here going on where, and let's start down here. So this is our tRNA. It has these 3 exposed bases. That's its anticodon. This is an amino acid. We need to pop add some ATP in here. And then we form this molecule here. This is our charged tRNA. The word charged means it carries an amino acid, AA, plus there's a high energy bond. So this black bond there, that's high energy. So basically taken the energy from ATP, I've broken off a phosphate, and I've put that energy into that bond there. Okay. Just while we're on this, just in terms of anticodon and amino acid, it's the anticodon causes the binding of a specific amino acid. And yeah, there's enzymes involved. So yeah, so there are 64 different anticodons. So there's 64 possibles. And amino acids, there's 20 possibles naturally occurring. So that's where, there's numerous anticodons can code for the same amino acid. So that gives us our degenerate code. And we get 64 possibilities because I've got 4 possible bases. I'm using them in threes. I've got 3 bases in the exposed bases of a tRNA. So therefore, yeah, that 4 to the power 3 is 64. That's how we get to 64. And while we have a 3-base coding system. So in the cytoplasm, the tRNAs bind specific amino acids. using energy from ATP that's ultimately come from respiration. And therefore, I've now got these charged tRNAs that can diffuse to the ribosome, and then we're going to undergo translation. So here, let's pick a decent colour. I do like a little bit of purple when I'm. So we're going to focus on this bit here, the interaction between a tRNA and a ribosome. So this is going to be our translation bit. So what do I write for a translation answer? Well, I'm going to write first of all that the mRNA is bound by ribosomes. It isn't just one ribosome, there's numerous of them. They all just jump on and start. Okay, and then what happens is that we get codon anticodon pairing, but it's important that you can show you know what those words mean. So I would always say something along the lines of, that a tRNA anticodon forms H bonds. Again, I'd write hydrogen in exam with bugging the word specific, it never hurts, specific codon or complementary codon on mRNA. So codon is a mRNA word, whereas anticodon is a tRNA word. Okay, and any excuse to name bonds, name bonds. Okay, so that's going to mean that the right tRNA binds onto the mRNA, and this is within the ribosome. And then another one comes in, another tRNA enters. I've already said about anticodon-codon pairing, so I'm not going to repeat myself. And the ribosome joins the amino acids by names of bonds get marks by peptide bonds. I'm forming a polypeptide. So that then happens. Let me just zoom out a bit, create some space. So where are we now? Right. So we've done that. Next, the ribosome has to move. And this is where it's important that we say how far it moves. So the ribosome then moves. Ribosome moves or advances one codon. Therefore, it's a non-overlapping code. And it keeps, and the process repeats of tRNAs coming in, leaving, peptide bond formation, and then the polypeptide is released. At a stop codon. The stop codon, there is no encoding tRNA, so tRNAs don't exist with a corresponding anticodon. So the ribosome waits around a minute, no tRNA arrives, and therefore it just dissociates. So the ribosome will, the top bit falls off, the bigger bottom bit falls off. So that kind of bit goes, this bit goes, and it will release the polypeptide, which, as you can see, is already folding into a protein. So that's why, yeah, protein is the thing. It's a folded polypeptide, or the final product. So that's translation. And just quickly, because I've not hit 15 minutes yet, I put it here about non-overlapping code. Remember, you have to know that the DNA code is non-overlapping. Well, that's because the ribosomes move a codon at a time. They don't just move one base at a time. We need to know there's a degenerate code. Well, that's the bit down here where I've got 64 possible codons or anticodons, and I've only got 20 possible amino acids, and therefore there's numerous anticodons or codons code for the same amino acid. So that's degenerate, and then final bit of the code is that it's universal, and that just means that I can put, I can take a gene out of here, I can pop it into another eukaryotic cell, and that cell will still make the same protein. So all life uses the same codon system. AUG in the mRNA, AUG anticodon always codes for the amino acid methionine in the whole of life, apart from some really primitive bacteria. So hopefully that makes sense. Again, it's focused on names of bonds. Any name of enzyme, mention it. It's last thing I'll say is it's really good for essays. Is this? It's good to to understand a lot of topic 8. It's important. It's also crops up as 5-6 markers, describe transcription, describe translation, but it's also really useful for essays because if it's a movement essay, I've got ribosomes moving, I've got mRNA diffusing out of the nucleus, I've got tRNAs in a cycle diffusing to and away from the ribosomes. So it's really good for movement. It's really good for cycles because I've got a cycle here. The mRNA incidentally gets broken down, and the mRNA nucleotides will diffuse back in and get rebuilt. So there's like a cycle there as well. It's good for energy transfer because it requires ATP to load the amino acid onto the tRNA. Again, a lot of people think that the ATP is needed directly at the ribosome. It's not. It's needed before the tRNA gets to the ribosome. So it's really good for energy transfer. And it's also really good just for the impact of mutations. That if there's a mutation, how it can lead to a different characteristic. Well, this is the mechanism. A mutation in the DNA calls for a different sequence of bases in the mRNA, therefore different amino acids bond, and you can go through that detail, and therefore you get a different shape of polypeptide, and therefore that protein has a different function that might cause a different characteristic. Wonderful. 17 minutes. I'll stop there.